first of the four major steps in creating ATP is called glycolysis. Glycolysis begins in the cytoplasm with an input of one glucose molecule and, ironically, two ATP molecules. This ATP splits the glucose molecule in half, forming two pyruvates. The two ATP that were used become two ADP and two free phosphate groups, or P. These products create 4 ATP and frees up 4 electrons. These 4 electrons are then transferred to the 2 electron carriers called NAD+. With these electrons now attached, NAD+ turns into NADH. These NADHs are then stored for later in the process. The second major step is called pyruvate processing. This is the stage in which the process enters the mitochondria. Pyruvate processing takes place in the matrix. In this step, one carbon is removed from each pyruvate and released from the body as CO2. The remainder is called acetone. Then the electrons released in these reactions are collected by electron carriers, NADH, which become Na. Electron carriers NAD+, which become NADH. Once again, these carriers are stored for the electron transport chain. Lastly, the remainder of the pyruvate attaches to a coenzyme A, and together they form acetyl-CoA. After acetyl-CoA has been produced, it enters the citric acid or Krebs cycle. The chief purpose of this cycle is to break food down to make energy or ATP. During the process, acetyl-CoA generates energy, most of which is transferred to the electron carriers NADH and FAD as protons, therefore reducing them to NADH and FADH2. There are 6 NADH+ and 2 FAD reduced in this reaction. These are also stored once they have all of their electrons inside of them for later. 1 ATP is also produced per turn of the cycle, forming a total of 2 ATP. Acetyl-CoA is further oxidized, forming carbon dioxide. Water is also a byproduct of the reaction. The final step of cellular respiration happens in the mitochondria's inner membrane. The electron carriers, NADH and FADH2, donate the electrons to proteins that act as electron receivers. With this donation of electrons, hydrogen ions, or protons, are pumped through these proteins into the intermembrane space. After passing through a few proteins, the electrons attach to hydrogen ions and oxygen to form water. The next part of this stage is chemiosmosis. As we've already seen, there is now a concentration gradient. The intermembrane space has many more protons and therefore is more positively charged. So, the protons want to diffuse back into the matrix. However, they can't go through the membrane and so need a proton to let them through. This is where ATP synthase comes into play. The protons diffuse back into the matrix through ATP synthase. This is called chemiosmosis, and the protons turn ATP synthase almost like a generator. The physical transformation of ATP synthase as protons move through it synthesizes the inorganic phosphate and the ADP to form ATP. Thank you for listening, and remember to